So I'll just introduce myself. I'm Frances Elmsley. I'm a clinical geneticist at St George's, and I'm clinical director for the South East Genomic Medicine Service Alliance. And I'm really pleased to welcome people to this, our autumn festival. We've held spring and autumn festivals since the inception of the GMSA. I'm not sure what number this is, but it must be about our 8th festival, something like that. And I'm really delighted that we're talking about metagenomics today. And I have to confess that I had to ask my colleague not that long ago what metagenomics was, because I'm a clinical geneticist, but I now know very well. And actually, one of my colleagues asked me that question the other day. So I was very pleased to be able to tell her what it was. So welcome, everybody. And we're going to start with Professor Jonathan Edgeworth, who is the director at the Centre for Clinical Infection and Diagnostic Research at St Thomas's Hospital. And Jonathan is going to talk to us about what is metagenomics, so you're answering my question, and why is it exciting? So over to you, Jonathan. Thank you very much.
Thank you. It's a real pleasure to be talking here, and actually normally obviously talk to infectious diseases folk and microbiologists, whereas today it's a fantastic opportunity because I assume we have cancer geneticists and human genetics experts here. And actually, Francis, the notion of what is metagenomics, every now and again I go, so what is metagenomics to AI, just to see what the latest definition is of as well, because that's important. So I will be speaking with a sort of human genetics audience in my mind, but I know there are some infection colleagues here as well. And actually, I think hearing about it at the interface between disciplines as a perspective is useful. And also, what is it and why is it exciting? Well, I think, why don't I just say what we're doing, and then along the way explain what's novel and interesting and exciting about it, so it's like a case study. So so clinical metagenomics is you take a clinical sample and you get rid of the bit that you're all interested in, i.e. the human cells, by one way or another, chemical or mechanical, just to leave all of the microbes behind. You then aim to sequence all of their genomes. Of course, they're much smaller than the human ones, aren't they? You know, a few thousand bases to a few million bases. And then that tells you what's there, what virulence genes it carries, what antimicrobial resistance genes it carries, and what variant it is, and whether it's novel or unknown. So essentially, in theory, you can see everything in a clinical sample that you need to know. If you can do that in like 6 or 7 hours, you can provide clinicians in an acute care setting with comprehensive information about what's going in on those samples for them then to decide what's important and what to treat. And that is a totally transformative proposition, because actually, to some extent, it's quite easy at the moment. You don't know what the bug is on arrival. You know, you can see the CT scan and the biochemistry and all the other labs, and you get the clinical details. And then you make an empiric choice on antibiotics. Empiric means your best guess, because you don't know what actually is causing it. And then you have standard antibiotics that are used. So imagine after having done that all your life, suddenly it's like, well, there's no guessing anymore. This is what's in there. That's actually quite a challenging proposition, because you then have to decide and you have to target rather than just giving broad spectrum. So that's quite a behavioral journey for clinicians, but an important one. Because if you target with the right antibiotics, the patient will do better. You also won't have the collateral damage of just treating a whole bunch of stuff that then just drives more resistance. So that's why it's key. So look, this is representative of our team. It's not everybody. We're about 15 odd people now, actually. It gives you an idea of the breadth of folk. We have a couple of bioinformaticians and research scientists. Simone Jelinas is our program manager. Vicky Ennie has moved to the Ellison Institute a month ago. She was our research-focused person, and Gul is doing a PhD down at King's. So I have to refresh the pictures here. I'm going to go through the vision. I'm going to talk about the lab stuff. I'll then talk about how we've gone from a single-site implementation into a network of excellence program, and then look to the future. So next slide, please. And just to say, we start off by doing research that requires ethics. But then, as I said, when we go into a service evaluation setting, I'll talk about that. We do that under local governance framework, so it's not in a consented process, it's a service. And I also say I stopped my clinical work at the beginning of this year, actually, and I've joined Oxford Nanopore actually in 2022 on a part-time basis of VP Medical Affairs to strengthen the collaboration between industry and healthcare. We get our funding from various places along the way. Next slide. So, microbiology, you know what? it's not the most forward-looking profession. In fact, generally, when I took over from my predecessor, Susie Eakin, she said, have any book you want on the shelf. And so I picked a few up, including this one here, and put it back on the shelf for 20 years. But just opened it a while ago and saw some wonderful quotes here. Look at this. Sir William Osler, as far as and they were mostly microlabs in those days, are concerned, I'll not say they're out of date because they never were in date, but I'll say they're shockingly behind the times. Well, if he came back now, 100 years later, he said, what are you still doing? Mashing up horses' hooves and putting it into sheep blood agar and growing the bugs on the plate? That was out of date when I was around. So it's quite understandable, isn't it, that at the bottom quote there, that often you'll find microbiology laboratories in some ill-equipped room in the cellars, not like our genetics and cancer genetic laboratories that usually have plush new floors and in new buildings and the like. So I think it's important, if microbiology wants to join the fourth industrial revolution of genomics, AI, and digital healthcare, it needs to get itself away from culture. Of course, there's PCR tests as well, but they're quite limited. But culture's the core of what we do. But that's not easy because this is firmly entrenched culture. Again, it's what we're all used to. I know it's slow. I know it doesn't pick everything up. But it doesn't cost a lot of money, actually. And it is embedded in our mindsets. So the transformation to move people away from these culture plates is pretty huge. So I think the first thing you need to have is a really compelling reason why we need to do that. So next slide, please. And to try and get the sort of funders and, government and academic organizations and the like interested is to bring infectious diseases together, just for everybody to realize what a huge challenge it is, and really use COVID-19 as a hook to just illustrate just what a huge human and economic impact that can be. So on the left-hand side there, you've got high priority pathogens, and each one of those could keep you going for your whole research career. In fact, some people become a doctor, just TB doctors. Each one of those is big in its own right and causes huge morbidity and mortality. But then you walk on the wards, and it's sepsis and respiratory infections, it's syndromic diseases that again are big killers. If you bump into the infection control doctor, they'll talk to you about hand hygiene and isolation and hospital-acquired infections. And of course, because, as I said before, almost always when we're treating patients, we don't actually know what they have on arrival. We have to give broad-spectrum antibiotics, and that drives antimicrobial resistance, predicted at current rate to be one of the biggest killers globally by 2050. And then on top of that, of course, we've had COVID-19. Viruses are spreading rapidly, increasing their geographical range from chikungunya and dengue and Zika and the like. And, we are under threat potentially of another pathogen pandemic, H5N1, and even in a sort of nefarious geopolitical situation we live in now, the potential to synthetically create organisms that are more virulent than the ones that nature can produce herself. And again, so clearly, culture ain't going to protect us from this and our current way of doing things. So the counterfactual of, metagenomics, I'll come to it in a minute, it's really difficult. How we're going to do it is what? So you want to carry on trying to deal with all of these issues with culture? Of course we can't. And so that's realizing that, I think, makes us need to redouble our efforts to seize the future. Next slide, please. So you need a vision, actually, that moves on from just scaring people. Next slide. And so the vision that we've set up, and Anna, I think, will talk to this too, because we're working together with UKHSA, is if we can embed metagenomic sequencing, so that's a sort of 6-hour protocol that takes a clinical sample into a genetic report of what's in there, if we can embed that in our clinical laboratories to provide those results to the intensive care unit, so you start with our sickest patients, give those results to infectious diseases and clinical microbiology teams as well as the intensivists and our infection control and pharmacy team, then that will help them embed that in practice and start realizing the benefits. So there's a clinical value there. At the same time, of course, because genetics is digital, the digital genomic sequence can be sent anywhere and kept forever and analyzed in many different ways. Send that data in real time through to Public Health, UKHSA, so they can construct genomes and sift through unclassified reads and give us that early warning of emerging and novel things. And then the UKHSA can feed back to us, to those ICUs, if something unusual or novel appears. You can also sort of put an overlay of biosecurity and academic discovery on that as well to advance. So that was our vision of what we were going to create. Next slide, please. The question then is, okay, I've said in the ICU, so we can't just have that in a few great centres around the country. How do you make that a proposition that's equitably distributed around the UK? So we have about 285 UK intensive care units in the UK. We're not going to immediately be able to jump to that whole network, but we need to have it in mind. There are, however, 12 specialist respiratory centres, high consequence infectious diseases units and ECMO centres. And the high consequence infectious diseases units are where they would admit anybody with a highly virulent novel emerging pathogen. And these are the sites around the UK on the left in red there, where those patients would go. And then there are ECMO units. Now, this is if somebody fails conventional ventilation, you take the blood outside the body and oxygenate it in a box and essentially keep them alive while their lungs repair. And these are specialist commissioned units, and they too are around the country. So I did most of my clinical practice over the last 20 years on the ICU, including an ECMO HCID unit. And so that's what prompted me to want to focus on a metagenomic test for severe respiratory failure patients coming into this unit. As is almost always the case, isn't it? If you're doing research or innovation, focus it on the things that you see day in, day out yourself in your hospital. That helps to make it most productive. Next slide, please. So that's our target, the ICU network in the UK. But of course, first of all, you've got to do it at a single centre. So how are we going to do that? So this was retrospectively overlaid on what we did. In other words, it's only after we made the advance that we looked back and said how we did it. But it fell into 3 phases, basically. And that was you develop a metagenomic protocol with research scientists. You validate it with your clinical laboratory as they would validate any new novel test. And then you go into a service evaluation in your intensive care unit on patients. So the intensivists identifying patients who've got severe respiratory failure, they don't know what the infection is. They do a deep wash of the lung. Actually, they were putting the samples in the fridge. We picked them up in the morning, did a metagenomic sequencing test. And on the ICU in the afternoon, I went down. I don't do that anymore. My colleagues do, and provide that metagenomic result at the bedside. And you record what the intensivists do. After a period of time, usually over winter, stop the service, look at the results, see what improvements are required, redevelop the assay, revalidate it over the summer, and then go back out into service again in winter. So you have a continuous improvement cycle. We've gone around this 3 or 4 times now to get towards the protocol that actually works. Because you can't just, with your own genetic tests you do in any discipline, it's not like commercially available protocols and boxes. These are things that have to be developed by clinical scientists. We view that if we can make progress through this cycle, we'll get to the point of something that can go into ISO accreditation. Next slide, please. So we did this first with the 16S test. This is just detecting the bacteria. And I think I'll pass over this quickly just to say that this confirmed to us that we had a mechanism of developing sequencing tests where you use the nanopore sequencers that give you real-time output so you can actually condense a workflow into 6 to 7 hours. Next slide, please. We then went on to our first metagenomic test that was actually developed initially in University of East Anglia, and they were lysing the human cells by putting in a chemical detergent and it binds to cholesterol. They then lyse all the human released DNA and RNA and then extract the microbial DNA. We validated during the COVID pandemic on clinical samples from patients in a research setting to prove that we had good performance characteristics, sensitivity and specificity for bacteria and viruses, confirm that we could identify antimicrobial resistance genes. And also, this is the beauty of it, if you sequence the bugs, I mean, we do a report after an hour of sequencing, so on the same day for a clinical decision. But if you carry on sequencing overnight on the MinIRN flow cells, you get enough data to be able to construct a whole genome. And then you can then compare the genomes of bacteria in different samples to identify outbreaks. That validation worked, and then a year or so later, we went into a service evaluation period. Intensivists changed antibiotics in half the cases. We found some interesting unexpected organisms in about 10% of cases. The thing about that first workflow was, next slide, however, that it doesn't detect RNA viruses. So we've now been on this third implementation cycle where a PhD student in our lab developed a mechanical way of blasting open the human cells that then preserved all of the microbes that you'd expect in a clinical sample. It was an easier workflow to do, takes 6 hours, and we were getting good genome recovery too. And last week, open access, we just got this paper published in Lancet Microbe showing that the service was valued. It was good for antimicrobial stewardship again. And again, we found this 10% of samples had hidden and unexpected organisms in them. Next slide, please. So this basically, I will just go into some of the lab work just to be clear about what it is and where we are now. And it takes 6 to 7 hours. So you do the mechanical depletion, you extract. Because you're detecting RNA organisms, you have to do a reverse transcriptase step to do single-stranded cDNA synthesis, PCR amplification, sequencing on a gridiron, internal spiked positive controls into each sample to show that the workflows worked, negative and positive controls, thresholds for reporting, and automated production of reports. The first one comes out a half hour, we clinically report 1 hour, and we do our final one after overnight sequencing. Next slide, please. Now, I think I do wonder in my mind, unlike a lot of human genetics, actually, we work on respiratory samples that are pretty disgusting, heterogeneous range of samples from, some of them are clear and colorless BALs, but they're bloody, they're pussy, they're mucousy. And an enormous amount of the challenge in this discipline is going from this heterogeneous real-world set of samples into clear, colorless, 0.5 ml of nucleic acid. And that's what quite a few people have done PhDs on, including Adela. And so that's the first step. And then at the back end for bioinformaticians, next slide, please, a lot of it is into the bioinformatic pipeline. And this is something that currently Dan Ward has developed. It is an SSD, a 5 terabyte mobile phone size device that we fit onto the back of the gridiron, doesn't require web access. It contains, pangenomes, human T to T genomes, contains 2 microbial reference databases, FDA Argos, and another. And essentially, it scrubs the human reads and bins them against those reference human databases. And then what's left is mapped to the human genome using Kraken, a centrifuge, and then you get a clinical report. It takes 5 minutes to produce a report. There's also a BLAST tool on board that now automatically analyzes those organisms above threshold to give you confirmation that you've got the right bug. And at the same time, and this is where linking through to Anna's talk, there is an ability in real time, i.e. same day, to take all of that metagenomic output when you've removed the human genes, human reads, and then send that data to public health so that, again, thinking through to the vision, you could have ICUs dotted around the country in real time, sending metagenomic data from the respiratory tract of people with pneumonia up into a central repository that could curate, quality assure, and then analyze, looking for novel and emerging organisms, or indeed even something like, the emergence of a new antimicrobial resistance gene. And because, if it happens in one site on one day, you might think it's just a single isolated event. But if centrally you could see, well, there was one coming from Newcastle, another one from Manchester, and next week there was another 5 to 10. You would say, look, actually, we're starting to see something big here kicking off. So that's the benefit of that part. Next slide, please. So I'll just give some examples of this now. So this was a few weeks ago, an interesting one. Chap with acute infection, green sputum, septic shock, failed ventilation, ended up on ECMO. On arrival, bit of Klebsiella in the chest. Is that what caused the pneumonia? Hard to say. Early abscess formation on the CT scan, went through all the antibiotics, didn't improve. On the 7th day, repeated that washout of the lung and nothing grew. But the metagenomic report here, 3,000 base pair average reads, Klebsiella pneumoniae, 80,000 reads in that sample that didn't grow. So we got a median of 70-fold depth. Didn't completely close the genome, but got pretty good coverage, and could see that it was a hypervirulent ST66 with lots of virulence factors. And there's the Klebsiella pneumoniae here, bit of herpes simplex and tobacco mosaic virus. So again, there's that ability to reveal things in culture negative samples, but not just tell you it's there. You get the whole genome. Next slide, please. And now I think I'm just going to show a couple of, yes, first of all, just to say, we could talk about this. Actually, if you do it on the right patients, severe patients with respiratory failure, and particularly from the community, the thing about metagenomics, just like with rare diseases, each one of your rare diseases are rare, but you bring them together, and I don't know, what is it, 1 in 20 might have something, so it's sizable. You know what? It's exactly the same for infectious diseases. There are a lot of rare, unusual things causing pneumonia. There's a long tail that individually are rare, but when you add them up, it's about 10%. And this is what's going to really do it, I think, for the pull and the case for bringing metagenomics in, which is basically letting people see that there's so much going on that's invisible, that's important. So hypervirulent bacteria with virulence factors. This was a disseminated HSV2 infection in a post-op patient that was causing liver disease and was obtunded. Antimicrobial resistance genes you're not expecting. Tropharema whippley is an interesting one. Neisseria meningitis in a patient coming from Africa. Fusobacterium necrophorum disease, atypical mycobacteria. We had a TB, parvovirus. The list could go on. Next slide, please. I will, however, give a couple of examples. And the first one here is somebody that's coming in for cardiothoracic surgery, 59, initially is extubated and doing okay. CRP goes down, but then goes up again, reintubated, penicillin allergy, so was put on gentamicin and vancomycin. And I was there with the intensivist and said, look, let's do metagenomics. And then the next day, respiratory metagenomics, Legionella pneumophila, which was a huge surprise for the clinical team, confirmed by PCR, treated appropriately, and the patient did better. But then took water from the taps around the bed space and found that the sink next to this patient had high titers of that exactly same sequence type of Legionella. So they serious incident declared, filters on the taps, and presumably prevented other cases. Next slide, please. And this is just one more case here that happened about 6 months ago. 64-year-old chap, community-acquired pneumonia, spent a long time in hospital, had a PE, then a bit of a hospital-acquired pneumonia. They did a CT scan, found this necrotic subcarenal mass. They thought it might be TB. Auramine and TB culture was negative on sputum. They even did an EBUS, an endoscopic aspirate of this mass, but it was auramine negative, culture negative, and the histology didn't show TB, so they thought it was cancer. We're about to go for an open thoracic procedure, actually, to biopsy, and deteriorated just the day before going, so they stabilized on ICU, did respiratory metagenomics, we found mycobacterium tuberculosis. So, that stopped somebody going to theatre when they were, which would have all the devastation of potential for transmission. Next slide, please. Good, I hope now, yes, so basically that's all the single centre stuff. So we showed this to NHS England, and just like many of you on the call will be part of Network of Excellence schemes, we joined with Southampton and Central and South Genomics unit to have a severe presentation of infectious diseases group. And we sold them that vision of, well, if you put this in centres all around the UK and sent the data to UKHSA, that would give you benefits for patients, and it would give us protection against, media and government pandemic warning for pandemics. So we have started that in 23, and we got Network of Excellence money, but then the funding for this has been expanded by Department of Science, Innovation and Technology for £30 million, so up to 30 sites actually around the UK. So the pilot in these sites has gone sufficiently well that the case has been made to create a truly national network, including Belfast, the devolved nations, Belfast, Cardiff and Edinburgh. Next slide, please. So we provide, Guy St Thomas's provides validation documents and SOPs for reporting and the like, and we've got ethics for data integration sent to all these sites and an ability to improve the protocol along the way with sites. Next slide, please. This basically is now us overlaying that respiratory metagenomic capability on the 8 SID and the ECMO centres, just so you can see we're trying to show our movement around the country. And this was last year, 700, it was probably just before the summer, 700 data sets gone to UKHSA, and we've passed 2,000 samples processed across these pilot sites around the country. Next slide, please. So yes, that's it actually, and I think I am within 30 minutes, so I'll just take stock of everything, because that was quite a whirlwind, wasn't it, from the what do we need to single site to network. Just to say, Sharon Peacock, who's a leading light in pathogen sequencing, said in 2014 we need to bring microbial sequencing to hospitals. And then, a couple of years ago, Eric Topol says, well, what are you all doing culture in your micro labs still? So yes, we could spend a lot of time just lamenting the fact that things haven't progressed. So I suppose what's exciting and what we're doing here is saying, well, we're going to actually just do this now. So we've got a vision. I've talked about that of this dual use. The strategy is to implement it in micro labs up and down the country and build a digital genomic bridge with public health. We progress through learning, continuous improvement, incremental improvement at pace, recording the impacts, and we are finding unusual things, important things that have potential to transform care. And none of us are under any illusion about the challenges that remain ahead of us. Expanding the network, improving the protocols, applying to other sample types. We need quantitative standards. We need an EQA scheme. Guy St. Thomas is going to go into its ISO 15189 inspection at the end of the year. But we also need to generate clinical and health economic evidence for commissioners, international collaboration, as well as automating to be able to improve the sample flow throughput and ultimately regulation. So thank you very much for listening. I'll stop there.
Thank you very much, Jonathan. That's a really great overview of metagenomics, but also of the program, which I think is really exciting. We have one question in the chat from Kevin Frost, which I think you alluded to in your very last slide, which is how can we get this spread into district general hospitals, or will this be forever the domain of the big teaching hospitals?
No, it won't, but it shouldn't. It can't, can it? That's not aligned with what the NHS is about. So, 30 hospitals. In fact, actually, what's happened to pathology networks? There's hubs and spokes, aren't there? So actually, it might be, if you sort of think about the 25 pathology networks and you think about the connection of those micro labs that are often centralized, I bet you it's not logistically too hard to bring in district general hospitals. There is a drone service between Guy's and St. Thomas's as well now that's got Civil Aviation Authority approval to do a 2-minute sprint and looking to extend that elsewhere. So perhaps innovation needs to beget innovation. There has to be a willingness on this district general hospital sites to uptake it from the clinical microbiologists, from the intensivist. It's not a passive thing. You have to want it, and there's energies required to implement it. And I think actually, if interested district general hospitals come forward and say, well, how do we get linked in? What do we need to do? That will spur us forward to try and find the ways to bring people together. But definitely, it has to be a national network. Truly a national network.
Yeah, yes. And of course, funding is going to be an issue, but I'm assuming that you shift funding from one area into a different area. Moving money around in pathology labs must have some.
Yes, it has to replace. It can't be as well as. So there's that hard thing of most of the time, actually, we don't do culture anymore. There you go. If you really want culture, ask for it in addition. You have to replace, and that's going to be hard too, isn't it? Because our risk appetite has to be quite significant. Because obviously it goes back to, well, look, if you don't do this, well, what, we carry on with culture for the next 20, 30, 40, 50 years? We know that's not possible. So, yeah.
Yes, thank you. I should have said to people, please do put your questions in the Q&A rather than in the chat. And it looks like people have listened to me, so that's great. I saw, Amita, you had a hand up. Would you like to put a question in the chat, or did you want to ask the question directly?
Hi, Amita.
I saw your name on a paper, so I'm assuming you're knowledgeable about this.
No, sorry, that was a mistake. Beg your pardon.
That's fine. No, that's absolutely fine. I have a question, Jonathan, and that's about novel organisms. I'm just thinking, when you do the metagenomics, presumably sometimes you see a genome and you think, I've never seen that one before. How often does that happen, and what do you do when you're in that situation?
Yes. So, of course, if it's not in your database, you won't see it. If it's, the reads will go unclassified. So that's a possibility. And sometimes we see a lot of unclassified. And you go and you say, oh, it's just an anaerobe, or, oh, it's a bug we know about. We just don't have it in our database. And so you incrementally add things. Sometimes you see a whole bunch of weird names of things that are just commensal flora, quite frankly. So that's an easier one to address. Every now and again, you will see something unusual. So the Tropharema Whippley is an example of that. Whipple's disease, we all know that's an intestinal infection, but we see it in about 1 in 100, or perhaps even more than that, 1 in 50 respiratory cases, of course, never looked at for before. And we're seeing it now in a couple of cases that are quite possible that it is causing disease, but we're trying to work out how you sort of go through Koch's postulates to say, does Tropharema, particularly in a couple of pediatric cases, is this Tropharema Whippley actually causing an infection in those patients? And then another one was a whole bunch of unclassified reads at a hospital actually up in Newcastle in a patient with a bone marrow transplant who came in with an acute respiratory failure, and we blasted them, and it was Toxoplasma gondii, where we all know you can get toxoplasmosis. And so, but you have to be really careful. Every time you see something that's a little bit unusual and unexpected in the first time, you really interrogate the reads to be sure. And there are 3,000 bases, so it's a good match. So long as the hits are good and the bit score is good, we'll call it. But that's the sort of approach to new things.
Yeah. Okay. Thank you. We've got another question here. Sorry, I'm not seeing it properly. So this is from Jonathan Lambourne. How do you work out what's a pathogen and what's a colonizer or latent flora, e.g. HSV, as the airway isn't sterile, especially post-intubation?
Yes. So the first thing about how do you work out what's significant, that's just what infectious diseases microbiologists do with every lab result. Again, this is no better at telling you what is causing disease. If you did transcriptomics as well, that might add something that allows you to see whether the immune system is bothered by it or not. HSV is a big thing. So we have found cases where we're clear. You find HSV or CMV in the lung, you sort of repeat, you do a blood plasma to see if it's actually systemic, and then you have to assess it in its own right. And also Luke has done a Delphi process to go around and talk about clinical reporting guidelines to get some consensus about what you do when you find things like herpes viruses. So that is one of the definite ones, along with PJP as well, when you call it. So it's an interesting exercise, one to be done by consensus, but ultimately you get there just through familiarity. Once you've seen things a few times, that you fall into an understanding of how best to approach it. Although, for a while, there's probably a little bit more investigation of herpes viruses in the respiratory tract in ICU patients than you would normally do. But once you've worked through that, things settle down.
Okay, thank you. There are a few more questions have appeared in the chat. I'm going to just do a couple more minutes, but then we will go back into the next talk, and there'll be a bit of time for follow-up at the end. So just a question from Ula Mahadeva about what is the cost per test and how do you know, I think probably, yes, how do you know environmentally ubiquitous organisms like Aspergillus are not contaminants?
Yes. So it's probably running at about 130 to 150 pound a sample at the moment. So 6 samples on a MinION flow cell with a positive and a negative control. If we sort out automation, and we may move to the PromethION flow cells soon, we've got the R10.1 flow cells working. We may be able to bring the costs down further. And then was the other thing about commensal? Was the other thing about commensal the second part?
Yes. How do you know they are not contaminants?
Oh, Aspergillus and things. Yes.
Like,
Interestingly, it's not like a bit of Aspergillus falling on a plate in the lab in the air that you're not sure what it's there. So it's not a laboratory contaminant that we have never really seen in a metagenomic thing. Colonization, well, look, in these ICU populations, I think it's very hard. It's only at the end of a lot of excluding if you found Aspergillus in a respiratory patient, would you call it a commensal? You could do a galactomannan as well. There's other confirmatory tests. Of course, you can do a PCR. I suppose you have to think about that every time you do it. Is this real? I think it's fair to say that essentially whenever it has been found, it's been considered that it is significant, or at least the chance of it means that it'll probably get some empiric therapy. Now, whether that's for a week, 2 weeks, or not, it is dependent on each clinical case. But it is definitely another thing that's considered, and actually, I think that might be something we would add. We're also saying the same thing about Candida species. When do you report Candida, and when do you consider them significant? That's a little bit easier than Aspergillus.
Okay, thank you very much. I think we're going to have to move on, but Jonathan, there are quite a lot of questions in the Q&A now. They suddenly sort of started coming in. If you wouldn't mind having a look, and if you can answer some of them as we go along, that'd be great. But we'll hopefully have a bit more time at the end. And just a reminder to people, if they could put their questions in the Q&A rather than in the chat, it's much easier for us to monitor one thing rather than both.
Yep.
Thank you. So I'm now going to hand over to Professor Judy Brewer, who is the Professor of Virology at UCL and the Clinical Lead for Virology at Great Ormond Street. And Judy's going to talk to us about metagenomics for brain and CNS infections. And so over to you, Judy. Thank you very much.
Thanks very much. Can you see my screen? Sharing
yes, we can. Yes.
I'm just going to put it onto, just turn it. I'm just going to put it onto, is it now, hang on a sec, just putting it on, is that now full screen? Okay, yes. So thanks very much for the this lovely invitation, and great to hear Jonathan speaking about their experience, and of course we were one of the first sites to adopt the GSGT protocol, which I have to say was really interesting and informative. So I'm going to talk about a different application of metagenomics, and it's really to sterile sites, and like Jonathan, this came about because of the patient population that I work with. I work at Great Ormond Street Hospital, which, as you know, is a paediatric hospital, and about 40% of our patients are profoundly immunosuppressed, and they do get quite severe infections, which sometimes we cannot diagnose. And one particular area that was causing us a lot of problems were encephalitis cases, where you have a case of encephalitis, you test for everything you can think of, you have elements of inflammation there, and they're on toxic drugs, and you end up thinking, well, we might just as well give them immunosuppressive stuff just to see what happens, and quite a lot of the time that ended in disaster. So on the basis of that, we started a metagenomics service in 2014, and the aim of it was to identify undetected pathogens in sterile sites, initially brain and CSF. So sensitivity was paramount to exclude pathogens as a cause, therefore, and thereby enabling safe use of immunosuppressive agents. So we also needed to have good specificity. And then as a sort of adjunct to that, to provide surveillance for pathogens of high consequence, should we, detect them. So we carried out this metagenomics service, gradually improving it over the years. And in 2024, last year, we applied for ISO accreditation, and we were accredited by 15189 accreditation. And I think we're probably the first in the world to have that accreditation. So quite a milestone. And the service is accredited for tissue and sterile site. It could include blood, but we haven't at the moment optimized it for blood. We're just doing that at the moment. Since 2014, we now have done actually more than 500. It's over about 600 or 700 pediatric and adult samples. We have about 200 to 300 samples a year, 60% of which are not from Great Ormond Street. We have established a lower limit of detection for RNA viruses, which is more or less equivalent to PCR, perhaps a little bit less sensitive for RNA in any tissue or fluid. For DNA pathogens, either DNA viruses or bacteria, it's about 100-fold less sensitive than PCR. And we are aware of that when we carry out the test and adjust what we're doing accordingly. So we have audited this service, and we found a previously undetected pathogen in 25% of cases that we have examined, and that's higher if the patient is immunocompromised. In our audit, 75% of the results changed management, and that was a new treatment, withdrawal of treatment, and even the decision to also to change treatment. We're now linked to 3 national multidisciplinary team meetings, 2 in encephalitis at Great Ormond Street for pediatric encephalitis, and at Queen Square for adult encephalitides. And we've recently started an MDT with Moorfields, a national MDT for eye infections. And what we are particularly interested in about our method is it preserves host transcriptomics for predictive biomarkers. So just to give you a flavour of what we have diagnosed in our population, we have all kinds of things ranging from, I mean, mostly viruses in encephalitis, but ranges from, you know, things you might expect to see there to some of the pathogens that you really wouldn't expect to see, coronavirus in encephalitis. We've also had an avian paramyxovirus here, for example. In pink are those agents that have not previously been associated with encephalitis, and those that are starred are pathogens that we find only in the brain, and I'll mention that in a minute. So we've had at least 10, I think it's more now, pathogens that have not been known causes of encephalitis, and most recently we've also started doing quite a lot of hepatitis as well. I'll explain that in a minute. So I just mentioned that we see things that are not associated, we think, with encephalitis, and somebody asked, how do you know that? Well, in our case, we do very extensive additional investigations. We do immunohistochemistry wherever we can. We do electron microscopy, and we also do in situ hybridization if necessary. And we're fortunate that we're a lab that can do that. And I think it's really important that where you have a pathogen that's detected in this circumstance, where you're not expecting to detect anything, particularly if it's something completely unexpected, like a bird virus, Newcastle disease, where we actually did confirm it by immunohistochemistry, that you go on and do the relevant confirmatory tests. And what has happened with us is that we've actually now found that in 13 cases, not a huge number, but it's again about sort of 1% of the cases that we've looked at, we find the pathogen, or actually a bit less than that, but we find the pathogen only in the brain biopsy. It is not detected in CSF, stool, nasopharyngeal aspirate blood, or anywhere else. So that has actually changed the diagnostic pathway at Great Ormond Street and in recent years at Queen Square. And now we advise that you do metagenomics early on when you haven't made a diagnosis, if infection is still in the mix, and that if you don't find anything, you consider brain biopsy if infection could still be a cause before you start escalating to immunosuppression. And that is now really becoming standard of care at GOSH and increasingly at Queen Square. So just to give you a flavour of three of the pathogens, or three of the cases, we've got a lot of interesting cases. This was a case of a young woman who presented to Moorfields with a unilateral sort of uveitis. She was tested for every organism they could think of that might not normally cause uveitis. She did not. She had trials of antibiotics. Nothing seemed to help. She got worse and worse and worse. She was put on steroids in order to treat what they thought must be an immune-mediated disease. She had to have a cataract operation because she got cataracts from steroids. And she was actually getting to the point where she was going to have her eye out because she was worried that whatever this immune process was, that it was going to be a trigger the opposite eye having the same problems. Her clinician, Colin Chu at Moorfields, was very prescient, and he knew about our metagenomic services. I think he'd heard one of us speak. He sent us a sample of her eye fluid, and we identified leptospirosis santarosae, which is an environmental leptospirosis, and it's found in South America. And in 2018, she went on holiday to South America and went swimming in the Amazon. We were able to confirm the presence of this pathogen. You can see with very good genome coverage across the whole genome, and she was treated with doxycycline for 3 weeks, and her eye was saved, and she was fine. So here's another one that was a really weird case. This is a 51-year-old man, managerial job, keen walker and runner and climber, very active, very intelligent man, presented in 2020 with falls, memory loss, Parkinsonian features. He had oligoconal bands as his biopsy in his CSF. His MRI was inconclusive. Many infectious agents looked for, nothing found. Provisional diagnosis of autoimmune encephalitis, given immunomodulatory treatments, but continued to have progressive decline and progressive signs of encephalitis. In 2024, he had a brain biopsy. The histology confirmed that it was an encephalitic process. He was now being considered for cyclophosphamide. He was presented at the Queen Square MDT, and it was advised that we should do metagenomics. We only had an FFPE sample, but this is just to show that even on that sort of sample, you can find things. And what we found was dengue. So this was completely unexpected. Good, you can see the reads across the genome here, very good gene reads, confirmed by PCR in two different labs. Sequence was shown to be a dengue type 1, and the dengue cluster, the sample clustered with samples that came from Italy, Europe, France, and also South America, as you can see here. It turned out that his intrathecal antibody synthesis was positive. They could not detect any dengue in his blood or CSF. He'd had no foreign travel since 2015, no blood transfusions, no history of dengue or febrile illness. He'd only been in the UK in the last 5 years. Prior to his presentation, he travelled to France, Holland and Finland between 2001 and 2015, and he had travelled to dengue endemic countries, Southeast Asia and Trinidad, but not since 2001. So this is definitely a case of dengue. There's a similar report from the USA, where the patient presented with exactly the same symptoms and the same sort of chronic presentation that actually in this case was only diagnosed post-mortem. We wanted to know if this is the cause or the bystander, and we're waiting. Our colleagues at NIH who had the first case have got antibodies that they're now testing the brain biopsy with. I have colleagues in Brazil with whom I work. We looked at 30 brains from patients from this dengue endemic region who presented with conditions that were not encephalitis, and none of them were positive for dengue by PCR. So this seems to be a particular, that does seem to be likely that it's not persistence, bystander persistence. And then the question arises, so I think we're going to prove that it is the cause. And then I think the question also becomes, where was the dengue acquired? And could this be the first case of dengue in the UK? And what a bizarre presentation. So lots of questions, lots of pathogenesis issues to be unravelled in the future. And then finally, I wanted just to mention that pathogen discovery becomes a possibility with metagenomics, and routine metagenomics at that. Our routine metagenomics service was sent liver from pediatric cases that occurred in 2022 when there was a massive outbreak of hepatitis in children. It resulted in over 1,000 cases, about 400 of whom were admitted to intensive care units, and something like 47 emergency liver transplants took place within a period of about 6 months. Ourselves and others found the AAV2 virus when we did metagenomics. Our colleagues in Scotland showed that this was present in the liver by in situ hybridization in liver cells, and we showed that the liver was absolutely stuffed full of concatenameric AAV2 DNA, which we think is causing a toxic reaction that then leads to an autoimmune hepatitis. What was really interesting was that we also got involved in a case of recombinant AAV gene therapy hepatitis. AAV is the commonest vector used in gene therapy, and hepatitis is universal in these patients. The gene therapy guys know this. And the patients are given prophylactic steroids in order to try and prevent the hepatitis. We had one patient in whom the steroids were not enough. She presented with exactly the same toxicity in the liver, and we found the AAV2 vector genome concatenated again in the liver. Suggesting that the pathogenesis of AAV hepatitis may be similar. So this has led to a whole program of work that we're now carrying out. And just to reiterate that we're interested in the host transcriptomics, which are preserved by this method, because here we're able to do host transcriptomics on the wild type AAV infection, that's shown in red, and the gene therapy. And you can see here that the AAV gene therapy and hepatitis cluster together in the pathways that are upregulated, particularly innate immune activation and DNA damage response. And they differ from hepatitis B liver and from normal liver. So this has given us an enormous insight into pathogenesis and could, you can imagine, be used in the diagnosis in the future. So we're very happy in many ways with our method, but actually our method is one of last resort. Everything else has been done. It takes a long time to process the samples. It's an Illumina method, and it takes 5 days. And although it preserves the transcriptomics, we just feel that it's too long, and we want it to be more disruptive in the way that GSTT have done so with their metagenomic service. And just to remind you, the GSTT metagenomic service is much quicker. It is less sensitive than ours. But and it does also deplete the host genome and host transcriptome. So that's a disadvantage from our point of view. So in order to try and adapt the GSTT or the nanopore sequencing for sterile sites, we undertook a study where we made up mock samples of DNA and RNA pathogens. We used viruses because that's what we had to hand, but we've now shown the same is true for DNA bacteria. We made dilutions of those. We added in human DNA and RNA to simulate a high biomass sample, and we compared our untargeted Illumina sequencing, untargeted nanopore, and a capture method where you have specific probes to, in this case, over 3,000 viruses with Illumina sequencing. And what we were able to show is, here is the capture method. The capture method is incredibly sensitive, and even at the lowest dilution, which is tens of copies, is still giving you a good signal of detecting the pathogen that is present. Our current method is the orange one, and you can see that it is less sensitive. It does detect things at low levels, it misses some things, but it is more sensitive than the nanopore, the untargeted nanopore. So we were not able to use the untargeted nanopore to do this. What we were able to show is when you use a targeted method, you cut the 5-day turnaround, and this is all the aspects of the pipeline, actually down to just over 2 days. So late morning on day 3, having received the sample on day 1, you're able to report the result. And we looked at nanopore methods that were out there to try and use a targeted approach to incorporate the probes, but unfortunately the methods that were available required this conversion step to ligate the sequencing primers to the library, the targeted library. So we were not getting any advantage from the nanopore. So Oscar Torres in my group developed a method. I'm not going to go into it, we haven't got time, but essentially it allows us to link the capture library, which has the probes in and increases the sensitivity 100-fold, to the ONT sequencing pipeline with a simple PCR that takes about 5 minutes, and actually is probably now going to be incorporated into some of the existing capture library methods. And that means that you can you can sequence the targeted enriched sample, which is sensitive enough, which we hope is sensitive enough for us to detect things in sterile fluids by ONT, which obviously is faster. So how does the ONT enrichment fare? Well, here is his in yellowish color, and you can see that it's as good as the targeted Illumina. So the sensitivity now of the ONT method is bang up to date and bang up with the most sensitive Illumina methods. The methodology is faster because you're able to cut out this whole area here, which means by mid-afternoon or early afternoon on day 2, after an hour of ONT sequencing, you can report the first results from these sterile sites, so your high copy number. And then before you go home on day 2, this is receiving the specimen Monday morning, early afternoon Tuesday, you can report the initial results, and before you go home you can report these results. And the next day, day 3, a good half a day before the Illumina reports, you can report the final results. What does that do? Well, here you can see after 1 hour, you get the highest copy number, 60,000 copies per mil. And here, here you have also, you get the ones that are sort of medium high copy. You get some of the ones that are at lower copy number. By 4 hours, end of day 2, you've got almost everything other than the very most lowest level pathogens that are present, and by the next morning you've got the majority of stuff detected. So pretty good advancement on what we've already got at this moment in time. But what we've also done is we've looked at ways of improving the whole protocol, and we've looked at the pre-sequencing. The sequencing is fast, but the pre-sequencing bits are slow, and we've actually managed to shave things off: extraction, the cDNA synthesis, PCR optimization, etc., and we're down to this 7.5 hour protocol now, which, if you tack on an hour of sequencing, is now a same-day, highly sensitive protocol that can be used for sterile sites, including potentially for sepsis, which we're now beginning to look at. And here it is. This is the version 2. This is, the ONT one reports out about here. I hope you can see my pointer, but the R1 reports out about an hour or so later. But same day, it's now a sort of 8 to 9 hour protocol. That's the initial report, and then the more the sensitive, absolutely everything that's there gets reported, and the genomes get reported the next day. So I hope I've shown you that metagenomics of sterile sites has already changed. It's accredited in our lab. It's already changed the patient care pathway for encephalitis, and is now being increasingly applied to ophthalmic infections. We're working with Colin Chu at Moorfields to develop the patient care pathways for investigation of undiagnosed ophthalmic infection. The current Illumina methods are slow and expensive and last resort. The untargeted nanopore is faster but not sensitive enough, and our version 2 targeted ONT metagenomics enables simple, cheap, fast metagenomics with the potential for same-day reporting, i.e. potentially could be equivalent or near equivalent to the ONT untargeted method that's used on respiratory samples, where you don't need the same sensitivity. Our method has the advantage that it preserves the host transcripts, and we are looking now to incorporate transcripts that will distinguish between viral and bacterial infections, and also transcripts that will predict outcome in sepsis, for example. And the methodology is not only fast for metagenomics, but actually can be used for anything where you're using targeted enrichment. And of course, targeted enrichment is a mainstay of exome sequencing for, looking for rare disease mutations or cancer mutations. And this method can speed up just ordinary exome sequencing or cancer genome sequencing, for example. And what we now need to do is do what GST has done so elegantly, which is to develop a pipeline that can be picked up and put in another lab, an end-to-end pipeline, and that's almost done. We're getting there, and then hopefully to use the setup that GSTT has pioneered to try and see whether we can get this actually working in labs, as everybody would like, all over the country, so that this could become a potentially a first-line diagnostic test for sterile sites. And I'm just going to finish by thanking all the people, some of the people who've contributed to this, particularly Oscar Torres, Sophia Buttle, the UCL Genomics people, and Sophia Morfopoulou and Julianne Brown. Thanks very much.
Thank you very much, Judy. That was really interesting, and I'd love to see that progression of you getting your turnaround times down and down and down. An amazing piece of work. I was going to ask a question about the dissemination of this technology in the same way that Jonathan has managed to achieve, but with obviously a lot of funding behind it. I just wondered, is that something that will be coming, do you think? Have you got the Department of Health and Social Care or Office for Life Sciences interested in this?
Well, I think we need to do what GSTT did so effectively and come up with an end-to-end pipeline that can just be picked up and put in another lab. So we're working on that, and we have some funding to do that. We then are very fortunate, and then we have an accredited pipeline already. So we're going to trial it in parallel with that initially, just as proof of concept, rather in the, I mean, we don't need to prove that metagenomics works because we've already got that. And what we just need to do is prove that it's picking up all the things that our current service does pick up. So we've got archived samples and our routine samples, and we shall spend a couple of months or whatever just running them in parallel, and we have funding for that. The big question will be then whether we can get the funding to actually roll it out to beyond just Great Ormond Street. And theoretically, given that GSTT has pioneered the way with a nanopore platform, it should be possible to put this in the same way. But obviously that will depend on getting further funding and support, and we don't yet know whether that's going to be forthcoming.
Yeah, that's absolutely. It does seem a no-brainer really that this should be supported. There are some questions in the chat. One, I think that refers to both your talk and Jonathan's, but I'm sure you can answer this, is have you observed a reduction in antimicrobial use with metagenomics compared with culture and obviously alongside more targeted treatment?
So in our case, it's not about antimicrobial use. We're mostly detecting viruses in this. So that's not well. When we haven't looked at that, but what we have observed is a change in treatment. Contribute in about 30% of cases. Treatment was changed in the patient, and that might mean withdrawing antibiotics, broad spectrum antibiotics, and giving more immunomodulatory agents, and in some case starting antivirals. What we have observed is that it's stimulated a program of experimental antivirals, and we're having some amazing successes now. That's accelerated our program of antivirals, and we're just now starting to publish the cases where we actually are using new antivirals in difficult cases which have good CNS penetration. So it has stimulated that. But I think Jonathan is in a better position to answer the antimicrobial usage question.
Do you want to come in, Jonathan?
Yeah, first of all, Judy, I can't wait to try your method, actually, as you know, because I think that would be a great combo if we can both this, and then there's an offer for sterile sites and respiratory. So it's great having being able to collaborate, and also pediatric hospital and a predominantly adult hospital. It's really nice across the spectrum. So long may our collaboration continue, which of course is going stronger. Yeah. So just about changing antibiotics. So first of all, clinicians treat the patient, don't they? These are acutely unwell. At the bedside, you do not want to stop something ever in your life, really, that was essential, and the person, God forbid, gets worse or dies. So there's a lot of behavioural thing. In fact, there's a lot of the behavioural element around antibiotic prescribing is partly down different sort of people. Some people, they have the different balance, your tolerance of risk or uncertainty. But at least metagenomics can enter into that space. So certainly, if they wanting to stop antibiotics, they don't think there's something there, and you give them this result, that gives them the confidence to say, look, I can hang my hat to just make this final decision that at least the metagenomics was negative. Of course, if they still think they've got an infection, but the metagenomics is negative, you say, well, look, I probably will carry on the antibiotics. But I think where we're going to see the biggest impact actually is if for the first time you start showing clinicians what's in all of these samples, rather than having lived with just, well, I don't no, there's huge uncertainty. There could be lots of other things that we haven't grown or we haven't detected. They will know what the norm is. So I think they'll reset their mean point of decision down to not thinking, oh, I must add tazocin or meropenem and an insular fungin and vancomycin, but just say, look, we've done metagenomics on the last 100 patients with severe respiratory infection. All we found is the pneumococcus and the Haemophilus. There was nothing else in there in this group. Therefore, I can have more, being more comfortable at not reflexing to adding lots of other antibiotics. So I think it's an individual patient benefit in the right sort, but then there's that collective agreement on adherence to guidelines. It's a long behavioural journey, but it's possible.
Yes, I can see that. Thank you. And there's a question back for Judy. Is your fast V2 targeted ONT just for viruses at the moment?
No, it works. I mean, what you can detect is dependent on the probe that you have, the probes that you have. We were using viruses because we had the reagents all ready. We've now done it on quite a few bacterially infected samples and shown that it's pretty good. It's just as good as it is for the viruses. It's sort of pathogen agnostic. It's really about detecting the DNA or the RNA. That's the most important thing. Yeah.
Thank you very much. We will move on to our final speaker, and please do continue to put questions in the chat because we'll have a few minutes at the end for questions. So we are going to Dr. Anna Ari, I hope I've got that pronunciation roughly right, who is a consultant in infectious diseases and microbiology at the UKHS Health Security Agency and also Barts Health NHS Trust. Thank you, Anna.
Thanks very much. Hello, everyone, and thanks very much for the invitation to speak. So yes, I'm Anna Ayi. I'm a consultant in infectious diseases and microbiology, and I'm speaking today in my role in the acute respiratory infections unit at UKHSA, but I also spend 50% of my time working clinically at Barts Health. So I think Jonathan and Judy have given some really fascinating insights into the clinical utility of metagenomics, and I'm just going to give a brief overview of its utility for public health surveillance. So I'll talk a little bit about a sort of very brief overview of metagenomics, applications for public health, and then some of the challenges as well associated with that. And then I'll talk a little bit about how we're aiming to use metagenomics for public health surveillance, and a little bit about MScape, which is a study that I think Jonathan mentioned briefly. So next slide, please. So as Jonathan outlined, over the years, we've seen sort of huge developments in diagnostic capabilities, in particular in molecular techniques and sequencing. So we started off with culturing organisms in the lab on a plate, and for many years that was the only method of bacterial diagnostics, but as you know, that takes time. We then have PCR, which is a molecular technique which can detect organisms rapidly directly from clinical samples, and which has been in use in virology for much longer than in microbiology. And this identifies the organism by identifying particular targets in the genome, but importantly, you sort of need to know what you're looking for to use it. We then have whole genome sequencing, which sequences the entire genome of a particular pathogen, but again, you have to have identified that pathogen in order to do it. And then now finally, we have metagenomics. And when people ask me what it is, the only way I can describe it is that it's essentially whole genome sequencing of everything that exists in a particular sample. So all the bacterial DNA, all the viral DNA, and all the human DNA. And as Judy pointed out in that in answer to the last question, it means it's pathogen agnostic. So you don't need to know what you're looking for in order to find it. And it can importantly also be done direct from clinical samples. And it's really those two properties that gives it enormous potential for public health surveillance, which I'll come on to shortly. So because it sequences everything in a sample, it has the potential to detect novel pathogens that we wouldn't know to look for with PCR, for example. And it's also much quicker than culture, which can require days in the lab. And speed is obviously key to any public health response. We also know, as Jonathan pointed out, the culture is flawed and that certain organisms are very difficult to grow. So our view of the pathogens that cause human disease is actually very much biased towards the pathogens we were able to grow and detect. So it's that pathogen agnostic pathogen agnostic nature and the fact that it can be done from primary samples that gives it such potential for public health surveillance. Next slide, please. So I don't need to tell this audience that global health threats are increasing. With examples such as Ebola and COVID-19, we've seen how quickly outbreaks can spread. And traditional diagnostics often target one pathogen at a time, which can delay recognition. And metagenomics gives us a broader, faster approach. And crucially, it also gives us a system-wide view of pathogens across humans, animals, and environments, giving us that one health approach that we're so keen on in public health. So in terms of outbreak detection and response, one of the key things is the speed with which things can be detected and the fact that you can detect novel pathogens, as I mentioned. And that's obviously crucial in mobilizing a public health response. It can detect multiple pathogens in one sample. So for example, there was a recent increase in invasive group A strep in the winter of 2022, which was associated with influenza A, and previous seasons have seen concomitant increases in dual pathogens, and that can offer us opportunities to institute mitigation measures. It provides strain level resolution, so we're able to pinpoint transmission chains in a way that culture or PCR-based detection doesn't. And I think the future directions may allow sort of portable sequences to be used for on-site outbreak detection or integration with AI for real-time analysis and forecasting. And there's also talk about global databases and sort of data sharing platforms, which can allow for faster responses and for transfer of information between countries. In the field of biothreat detection, it can be used to identify engineered or unusual organisms in environmental or clinical samples. So it can act as an early warning system because all sorts of samples, air, water, soil, can all be sampled for biothreats or emerging risks. And importantly, metagenomics can detect genetically modified or unknown agents that might be missed by targeted assays, such as PCR. And there have been lots of examples, not necessarily in biothreat detection, but examples of environmental sampling using wastewater surveillance, for example, in the COVID pandemic. Wastewater surveillance detected new variants earlier than clinical sequencing in some areas. In the area of AMR, we can track resistance genes across human, animal, and environmental reservoirs, again, using that One Health approach. And it allows us to track the genes, a sort of resistome at population scale, and it captures both pathogenic bacteria and commensals, which, as I'll come on to, can be a complication. And uses might include early detection of rare or emerging resistance genes before they become clinically apparent. It can be used at sort of hospital level monitoring of resistance patterns or for trend analyses. So identifying if there are seasonal or geographic differences in resistance prevalence. It can also be useful in linking to antibiotic use. So correlating shifts in the resistome to prescribing data and agricultural practices. And again, future directions may include allowing us to integrate this data with national AMR surveillance networks. In the area of surveillance of endemic and neglected pathogens, it can allow for continuous monitoring of diseases with low reporting. So these may be things that are sort of present in low abundance, or they may be genetically diverse. So things like Chagas disease and leishmaniasis are very genetically diverse, and that can make detection challenging with traditional tests. And that's a gap that can be filled with metagenomics. It can also be used in sequencing ticks, mosquitoes, or rodent samples to identify zoonoses. And then finally, coming on to zoonotic diseases. So traditional surveillance often focuses on known pathogens in human cases, and that may miss spillover events that involve novel or highly divergent strains, or ones that are sort of asymptomatic or poorly symptomatic in the early phases. And of course, ideally, you want to detect the pathogen before it spills over into humans. And metagenomics allows us to monitor zoonotic pathogens at that human-animal environment interface, again, providing comprehensive biosurveillance. So for example, recently, metagenomic sequencing of bat samples has helped identify spillover risk hotspots for Nipah virus in South Asia. And that's detected asymptomatic circulation of the virus in bats in areas without human outbreaks. And it's also helped to identify spillover interface points like contaminated palm sap, orchards and or animal farms. It was also used recently in the influenza A H5N1 multi-state outbreak in dairy cattle in the US. So, for example, in the state of Oregon, H5 was detected in wastewater about 6 weeks before a poultry outbreak was detected, and about 7 weeks before wild bird H5 detection. And it was also used. H5 was also identified in wastewater surveillance, and that helped to confirm that milk processing plants were discharging material containing viral RNA into the sewer system. So importantly, that metagenomics can then identify viral RNA also from animal sources, not just human infection, and that can give us sort of earlier signs of viral circulation. Next slide, please. Now, of course, anything that has such huge potential will also come with some challenges, and I'm just going to talk about some of those now. So there are significant technical considerations. As Jonathan mentioned, samples may be contaminated or pathogens may be detected in low abundance, and that can make it difficult to be sure of their significance. Unlike a sterile sample, looking at environmental samples, that can make things quite difficult to interpret. And even if we're sure of their significance, interpretation can still be complicated. So, for example, with the wastewater surveillance I mentioned, it can detect viral RNA from animal sources and not just human. And that can be massively helpful, but it can, of course, be quite complicated to interpret if you're not able to distinguish between the two. I mentioned its utility for the surveillance of neglected diseases, but there are limited reference genomes for those pathogens. And reproducibility is also complicated, so different pipelines may give you different results on the same data. Similarly, with detecting novel pathogens, that's obviously, there's great potential, but it is also technically quite complicated to interpret unknown sequences, so it's not necessarily straightforward. Data. Metagenomics creates a vast amount of complex data, and that does bring significant computational demands. There's also a need for accurate metadata, which you obviously have in the clinical service, but for public health applications, that can be more difficult. And without that accurate metadata, it's quite difficult to interpret. Epidemiological interpretation can be quite difficult. So standardized lab workflows are really, really key to avoid sort of misclassification of pathogens. And the long-term storage and data management is technically quite demanding, as I'll come on to later, also quite expensive. In terms of ethical and legal considerations, so at present we share pathogen data across different agencies and even internationally to sort of protect public health. And there's a sort of implicit consent when human samples are taken for the purposes of diagnosing an infection in a clinical setting. But the complication with metagenomic data is that even though we take these steps to remove the human genomic data, we remove it as far as possible, there is obviously a concern that there is residual human genomic data there, which could be inadvertently shared. And that obviously raises ethical risks. So there is a real need, I think, for a sort of an ethical framework and standards and guidelines that will maintain sequencing quality and strengthen our confidence in the removal of human genomic data so that we can share that data safely and responsibly. And then finally, the operational considerations. So I think it's firstly important to highlight that there are different use cases with different goals for different settings, and it's important to consider those before starting because the operational considerations will be different. So the use case for surveillance of endemic pathogens in the UK will be different to that for outbreak detection in a low resource setting. And it's likely to need different sampling strategies, different pipelines, and different data sharing agreements. And so that's very important to consider upfront. And there's also obviously an upfront cost in setting up metagenomics platforms, and then additional costs in maintaining the infrastructure, and also a cost associated with upskilling the workforce and training the workforce. And those considerations are obviously even greater in lower resource settings. Next slide, please. So how do we hope to use metagenomics in the UK? Well, we hope that it'll give us a broader biosecurity net, helping us to keep pace with the evolving threat landscape. We hope that it'll allow us to detect threats and pathogens earlier, meaning that we can also institute public health measures earlier to reduce the impact. We hope to gain richer insights into particular pathogens, and that characterization of example virulence factors or AMR will allow us to make better informed decisions around countermeasures such as vaccines and antibiotic strategies. And we hope that all of this will provide value for money overall, providing a more efficient surveillance system. Next slide, please. So as part of this, we have implemented a pilot study called MScape. So this stands for Metagenomics Surveillance Collaboration and Analysis Program. And this is a pilot study which will ingest metagenomic data from a number of different sources. That includes public domain data sets, research cohorts such as SIREN, which is the study of healthcare workers, which was set up during COVID and is still ongoing. Data from NHS clinical services, including patients with severe respiratory illness, which is led by Jonathan and our colleagues at GSTT. And also samples from returning travellers, led by our colleagues at the Rare and Imported Pathogens Laboratory. And there will be other newer data sources hopefully coming online in the future, including environmental pilots using aircraft wastewater and samples from patients with neurological disease also. And as part of the study, the core analysis functions we're testing are large-scale processing of metagenomic data, so to allow surveillance of our seasonal and endemic pathogens, detection and confirmation of high consequence and high threat pathogens, should they be present. And we'll be looking at approaches to identify signals of novel, unusual or evolving pathogens, whether that's detection of the pathogen itself or patterns of other organisms present. And then finally, we'll also be doing further characterization of a smaller panel of organisms such as strep pneumo and influenza A to see if we can confidently identify things like virulence factors and AMR. Next slide, please. So looking forward, this is what we're hoping to achieve on our pathway to national surveillance. So an integrated surveillance system, which uses clinical and environmental samples, giving us the potential for sort of One Health sampling frameworks. And key to all of this is a health economic assessment. So what we want to do is evaluate the utility of metagenomics approaches as compared to our existing surveillance to see if and how metagenomics can add value to our existing surveillance. We also aim to develop an ethical framework which will allow us to share data safely and legally. And we'll also look to leverage our existing partnership with clinical and academic partners, as well as other government departments in order to accelerate the widespread implementation of metagenomics. And I think that's my last slide.
Thank you very much, Anna. That was a really comprehensive talk and really interesting alternative angle on all of this. I've got a couple of questions. I think coming back to the data issue, of course, we are storing increasingly numbers of human genomes, so massive amounts of data. And I suppose my question is twofold. The first is, do we need in sort of routine clinical care, once this becomes routine, do we need to store the data? Is there a sort of legal framework for storage of data on pathogens? And secondly, of course, you know, for your purposes, you would need to look at trends and you know, have that data available for to analyze trends, but how long would you need to store that data? So are we looking at a sort of increasing need, which could be massive need for data storage, you know, just in genomics in general?
Yeah, I mean, I think the answer is probably yes. I think there's obviously a difference between storage of the pathogen data and storage of sort of the human genomic data, and those will have different considerations and different sort of levels of sensitivity, if that makes sense. I think at the moment our protocol for MScape is really is quite limited. It's a pilot study, and whereas, you know, all the data is very much sort of locked down and not able to be shared at all. I think as you said, as more and more people start using metagenomics, it is going to become a consideration. And I think it'll come down to having a sort of data storage data storage plan like you do for other data. Like we just have to come up with one for metagenomics and decide how long we're going to store things for, what is reasonable, and what are the purposes it can be used for.
Yeah, thank you. I just heard a statistics last night. I don't know if it's absolutely correct, but I think it is, that now in Ireland, 20% of their electricity use is for data storage. And that compares with 4% in the US currently. But I thought, and that now it's become an industry. In Ireland, that's what one of their biggest industries is data storage. So I'm just thinking, God, the implications of that. But hopefully there are solutions coming down the track in terms of energy production and better batteries and all that sort of thing. And there's a question from Jonathan Lambourne. So for the centralized use of NGS, I think, and WGS data, does the entire pipeline need to be the same across all contributing centres? And if so, who decides what the right pathway is?
So you mean in terms of the MScape project, presumably? Is that the question, John? I think
he hasn't said specifically,
okay.
But I imagine if you're feeding in, I suppose if you're feeding into an external agency, does it have to be uniform?
I think there is a uniform protocol that is used, but not. I don't think every site is doing exactly the same thing. So one of the things that we're doing in the pilot is ensuring that we can be detecting the same things that the site has detected, if that makes sense. So we're still very much in the early phases of just being clear that we can actually detect what we think we should be able to detect. Yeah. I think moving forward, what we would want is some kind of harmonization across all the sites. But at present, I don't think that's a requirement at the moment for everyone to be using the same pipelines. But I would have to check that, John. I can get back to you.
Okay. And Jonathan, obviously, because you're going to have 30 sites, but they're working to your protocol, aren't they? All those sites?
Yeah, at the moment, we're focusing on everybody getting exactly the same protocol set up. So you could send round samples to an EQA scheme to say, are they all working well, to get standardization? And then as there are protocol improvements or evaluating new protocols, for example, Judy's protocol would then go through a controlled way around the network. That's the current plan, just because I think the voices around, well, if everybody goes off and does their own thing, you don't know where you are, and it could become chaotic. So at the moment, we're rather saying we all have to do the same thing, just so we keep something fixed, because of course the patient populations are different and the labs are different, and then also we haven't standardized the extraction platforms, for example. I mean, all the chemistry and the sequencing is. So there is still some minor changes. Of course, there's all heterogeneity in the sample types as well, the BALs, the NBLs, the sputum. So yeah, at the moment it's a single protocol, and I think on balance that's the right way at the moment. Yeah.
Okay, thanks. Judy, you wanted to come in.
Well, I just wanted to agree with the previous two speakers that I think initially, as we get used to metagenomics, and I think there's also the business of learning how to interpret, and every platform has its own quirks. And so I think people just starting out with metagenomics will need to sort of be guided and to sort of learn within the framework of what other people's experience has been. And so for that, it's probably easier to start with people using the same platforms, and then they can, they have common frames of reference. Because if one person's using an Illumina and one person's using a nanopore, they're going to have different quirks that you have to then account for, and so better I think at the moment. But I think that this area is now moving very, very fast, and I think a lot of companies have now cottoned onto the fact that metagenomics is looking like it might become a sort of disruptive technology, and I think that they're piling in, and so I think that inevitably, eventually, it will be like PCR, that we all started off doing PCR in the same way, and then different companies came in and developed different primers and different ways of doing it, and it'll be like that eventually, as we get used to it.
Yeah, thank you. Steve Copping has a question. With these rapidly expanding programs of activity, so this is for Anna, I think, are we confident we have suitable and sufficient biosafety and biosecurity risk assessments in place, including cyber biosecurity challenges and emerging threat vectors?
Hold on, I might need to read that. It's in the chat. With these
Yes, Steve Copping. It's in the Q&A.
Yes. I mean, that's a very good question. I think we are confident. I mean, we don't have, in terms of the data that we've got, we can't actually link our data back to any individual patient. So in terms of the sort of cybersecurity, that threat, we can't. The data is essentially anonymized. The only thing that would allow us to go back to the sites to tell them what we have detected so that they can try and find it themselves is if we detect a high consequence infectious disease, because we think there's a sort of significant public health requirement there for them to be aware. But even in that situation, all we can say is we've detected a high consequence infectious disease. You need to go and test for it now in your patients. So I think for the purposes of the study that we have at the moment, I do think we are, I'm confident, but I think that's the reason for it being a pilot, is to sort of evaluate all these things before it can be kind of implemented more broadly as for public health surveillance.
Thank you. So next question, Elizabeth Francis. How do you distinguish a real threat from background noise? Does detection of pathogen equate to a live or real threat? I think the answer is no, not necessarily. But can we determine the source of the pathogen threat? Who is that best for, Anna?
Right,
okay,
yeah.
Anna.
Well, I think my view is no, we can't. And I think one of the complicating factors is, for example, if we're going to be using metagenomics to look at our, for example, to look at strep pneumo surveillance, the sample type's completely different. In EMSCAPE, we're looking at respiratory samples. Our existing surveillance is all looking at invasive. It's all blood cultures and CSF samples. So the sample types are different, and I think the significance of strep pneumo in a CSF sample or a blood culture isolate is higher than it is in the respiratory tract. So I do think we have to. We just have to be aware of the sort of limitations, I think, and I don't think we are making any sort of, in terms of the surveillance, we're not making a statement as to the significance in a particular patient. We are looking at sort of the surveillance of the presence of the pathogen on a population level, if that makes sense.
Yeah, thank you. So interesting question from Tariq Sadiq. Would the panel comment on the ethics of sharing metagenomic results with patients, particularly around rare organisms or those of uncertain significance? Whether you do share, it'd be interesting to know, particularly in the age of increased access to online clinical information and patient anxiety, etc. Would that be Jonathan? Would you
I'm happy to. I was thinking of trying to answer that one by writing, so it's nice to say it. So in practice, a third of the time it's nothing. Like another 50% is just your usual stuff. Mm-hmm. And then that 10% of unusuals, they're unusual, or what we weren't thinking about, but we all know about them, the mycobacteria, group A streps, and viruses and what have you. So I think the frequency with which you'd find something. I mean, for example, there's another one where there was this HIV in the respiratory tract because it was disseminating. It was a new diagnosis. Of course, you do it peripherally, but I think that's all covered. We can do that, can't we? And of course, you don't give reports to clinicians with every single bug in it so that somebody can go Googling every single bug. You tell them Pseudomonas, Enterobacter, and usually the other thing, just to say, above threshold, it's usually just 1, 2, 3, and occasionally 4 organisms. It's not like, oh my gosh, suddenly I'm reporting vast numbers of different things. So these are the important questions to discuss, but the frequency with which it will actually become a practical issue, I think, is very, very, very low. We haven't had a single issue yet with anything that's been identified. But one thing that is on our mind, I admit, is Tropharema Whippley, as I said, Whipple's disease. The first time we found it in somebody who actually just had a thymoma operation, we thought, oh gosh, I Googled it, thymoma operation and Whipple's disease. We treated them with 2 weeks of ceftriaxone, and they were followed up by ID. In the end, we thought, no, it probably isn't it. There's a lot of literature about that. So that has to be resolved. So that's one example where that's got life, such that if you did report back, as I said, it's a paediatric patient, Tropharema Whippley. In fact, Judy might have done that on some of the patients as well. And then they look at it and say, has my child got Whipple's disease? And then, so yeah, hope that's enough.
Thank you. Judy, do you want to come in?
Yeah. I can comment because we've had quite a lot of these sorts of things happening, and actually the Tropharema Whippley was, I think the first patient was at Gosh, actually, I think. So I think what happens is that, I mean, what we need to stress, all of us, is that when you're using it for clinical diagnostics, there's a very stringent set of rules that we follow before we report an organism. So, I mean, you know, there's a one set of rules that GSGT have developed for the respiratory, and we follow those. And usually that sorts out things that we think are real and things that are not. And it's like anything. I mean, if you go into microbiology and you're faced with having to read a plate, an agar plate with organisms on it, you look at it, you think, well, what on earth is going on here? But with experience, people learn to work out, you know, they learn to know which colonies they need to test for, which ones are in high abundance, which one are in low, but what is important, what's not. We're doing the same with metagenomics, and those of us who've been using metagenomics have evolved these rules which allow us to determine whether what we're seeing is real. First of all, is it not a contaminant and not something that's just a bystander? Then in our case, if we have something that's completely unexpected, and we have had, we will go on and make sure that we confirm it by other methods. And we will do PCR, we'll sequence the virus, we'll do in situ hybridization, all kinds of things, because it's so important that we get that right. But in the end, if you've got something that's real, you have a duty of candor to the patient. You can't withhold real results from patients. They have the right to know what their results show. And if it's something very rare, but you're certain that it's there, and you're pretty certain that it could be contributing to their disease, then we do discuss it with the patient, even if it's a difficult discussion to have. I mean, in one case, I can give you an example. We had this pigeon virus in the brain of a young child who had a 16-year-old, actually not so young, who had an encephalitis that had remained undiagnosed for weeks and weeks and weeks and not responded to immunosuppression. Turned out that this child who came from the Middle East had a hawk who had killed a pigeon recently before he became unwell. No one had made the connection, and of course his parents felt incredibly worried and awful about the fact that they'd allowed him with an underlying immunosuppression to keep a hawk. But it had to be handled very, very sensitively. But we couldn't not tell them that this is where this is what this child had, and ask them about whether the child had any contact with birds and things. It had to be discussed because we had a duty of candor.
Yeah,
so
absolutely. Yeah, very interesting, your comments about working out what's significant and what is not, because it's completely analogous to, when now we do whole genome sequencing and our clinical scientists are filtering through all that data, and quite often then coming to the clinicians for our perspective on the variants that they're identifying. So it's, we've got our standard protocols and just the same.
And I think it is, it's worth saying that this is a team effort nowadays. We don't make these diagnoses on our own. We discuss them with our colleagues looking after the patients, and it's a back and forth. So, unless it's obvious, then then we exactly the same.
Yes, absolutely. So one question in the chat is there a plan to establish a national library to accommodate available data from different centres, or is the National Library sort of at the UK HSA? Just question.
So I think the answer to that is that we don't yet have the assurance that we can do that safely yet, if that makes sense. I don't think there are obviously various steps that are taken. There's sort of wet lab steps that can be taken to remove human DNA, and then there's steps in the sort of bioinformatics pipeline as well that can remove it. But I don't think we know that there is still human genomic data there in the data sets that we're analyzing. So I don't think the idea of having a sort of publicly available or nationally available data set at the moment is I think we're a little way from that yet. I think we need to be clearer about having a sort of an ethical framework as to what is acceptable to share, and and with who before we get to that stage. So at the moment, I don't think that's in the works at present yet.
Thanks, Judy.
What I should say is I think that's absolutely right. But I think what's happening now is more and more of us are talking to each other about what we're doing in metagenomics. And those of us who are already doing metagenomics actually there's a monthly meeting which is open to anybody to dial into. And it's co-run by Luke Snell and Sophia Morfopoulou. Sophia is at UCL and Luke is at GSTT. And basically people are invited to share their cases or their experience of using metagenomics, their case series, that kind of thing. And it's providing a lot of information around, use of metagenomics, routine use of metagenomics. So it's not actually sharing the data, but sharing the experience. And anybody's welcome to join that monthly meeting.
Fantastic, thank you. Could we ask you for the sort of link for that, and we could share it with this group? Is that possible?
Yes,
not right. It doesn't have to be right now. We can do it later. Right. I think
you can have it. Whether I can actually find it now, I don't know. But yeah, definitely.
Fantastic. So I think we're at the end of all the questions. So I would like to thank our speakers very much indeed. It's been really interesting as a human geneticist, absolutely fascinating for me in particular. And just for other people, we will be having a spring festival. We don't know what the subject is. If you've got any views on what the subject should be, please let us know. We do listen to people's views. And I think we'll be asking people for some feedback as well. But thank you very much, Jonathan, Judy, and Anna. Thank you.